So presenting for us today, we got Dr. Jae Kyu Ryu. He's the scientific program leader at the Gladstone Institute of Neurological Disease and associate adjunct professor at the University of California, San Francisco. And then I just wanted to make note before he takes over, he will need to get off the session. Um, Directly after his presentation. So if you have any questions, feel free to drop them in the chat and I'll make note of them and I'll reach out to him and get back to you. All right. Thank you so much. Passing it over.
Oh, thank you so much for kind introductions. I'm gonna share my slide. Okay. Thank you very much for the introductions. I'd like to thank the organizer for giving me this opportunity to present our previous research. Um, Today, specifically, I'd like to focus on the fibrin-targeted immunotherapy protect against inflammations and neuropathology in SARS-CoV-2 infections. For the disclosure statements, I have no relevant financial relationships, commercial interest, other conflict of interest. So, in the beginning of pandemic, it was unclear why patients with severe COVID-19 had such a high risk of blood clot. And clinicians observed the strokes, pulmonary embolisms, and the microvascular clots throughout the body, including lungs and hearts, and even in the brain, often in otherwise healthy individual. As the disease unfolded, it became evident that SARS-CoV-2 infection triggered not only respiratory illness, but also systemic vascular and immune disorder. So for example, coagulopathy, neurological complication in COVID-19 occurred during the acute infection and also persistent in long COVID. Coagulopathy affect the even young COVID-19 patients, including those with the mild and breakthrough infections. And we also found that the blood clot in COVID-19 patients resist degradations despite the anticoagulation treatments. And long COVID patient also features of the chronic vascular disorder characterized by persistent thromboinflammation. These slides summarize the clinical and experimental evidence supporting the thromboinflammation in patients with the long COVID. Overall, persistent vascular dysfunction appeared to create an imbalance between pro-thrombotic and anti-thrombotic mediator. For example, persistent vascular and endothelial dysfunctions marked by elevated VWF, factor VIII, alongside with the decrease in cleaving protease. And also the patients exhibit platelets hyperactivity and presence of the circulating microclots and a significant increase in monocyte platelets aggregates, especially in those with the symptom lasting over 12 months. Recently, there is clear evidence showing the complement system dysregulation is also involved in long COVID. Especially active long COVID patients show that the terminal complement imbalance, increased C5b, C6, and decreased soluble C7 complex, that indicating increased membrane insertion of the complex complements and tissue damage. So together, these changes can sustain thrombotic and inflammatory environments in long COVID. And this may contribute to both microvascular thrombosis affecting small vessel and tissue perfusions, and macrovascular thrombosis, which can increase the risk of larger vascular event. In addition, previous study have reported that the hypercoagulation and the fibrin deposit in COVID-19 are associated with the lung and brain inflammation. For example, hypercoagulability in COVID-19 is associated with extensive fibrin deposition in inflamed lung and brain. And the fibrin is derived from the soluble blood protein fibrinogen after activation of the coagulation cascade. It forms the central structural component of blood clots. Fibrin deposition after BBB disruptions, known to drive innate immune activation in autoimmune, inflammatory, and neurodegenerative disease. The second aspect of that, the make fibrinogen really attractive study was the unique structure function relationships. So what truly sets fibrinogen apart from the other blood protein is the coagulation factor. The soluble molecules circulating in the bloodstreams, but when the BBB is disrupted, the fibrinogen is converted to insoluble fibrins, forming the persistent scaffold protein that deposits at the site of the leakage in the brain. And this convergence exposed hidden inflammatory domain critic epitope that binds to CD11B subunit of complementary receptor 3. Another advantage of studying this pathway is that there are genetic tools available, including fibrinogen knockout and fibrinogen knock-in mouse model. So this allows to perform the loss of function study to understand genetically how depleting or modifying fibrinogen affect the neurological disease. So in our lab, using these approaches, we found fibrin is both necessary and sufficient to induce neuroinflammation and neurodegeneration across the multiple models of neurological disease. What we have found so far was that upon blood-brain barrier disruption, fibrin induced a dual role, pro-inflammatory and pro-oxidant response in microglia, polarizing microglia to neurotoxic cell phenotypes characterized by oxidant signature that coupled with the coagulation gene, leading to inflammations and oxidative stress, causing inhibition of repair, demyelination, and neurodegeneration, which are the part of the progression of the multiple sclerosis, traumatic brain injury, and Alzheimer's disease. The previous study have reported that the fibrin-linked neurovascular damage in COVID-19. This is the top left panels, the fibrin immunostaining compare postmortem tissue in COVID-19 infected patient brain. You can see that massive amount of the fibrin deposit in the brain. And this study, COVID-19 neurological symptoms, pathological alteration has been attributed to the secondary effect of systemic SARS-CoV-2 infection. And also, neurovascular injury, reactive microglia detected at the site of the parenchymal fibrin deposition in the brain of the patient with the COVID-19. And the bottom left, the functional MRI studies in long COVID patient without brain fog. You can see that with brain fog, patient with the long COVID show more extensive BBB disruptions. And this study clearly correlate with the brain fog in long COVID, and also increase of blood plasma fibrinogen is a predictive biomarker of the cognitive deficit after COVID-19 infection. However, the role of the blood clot in COVID-19 inflammation and neurological change remain largely unclear, and therapeutic to combat this fact are not readily available. So in this study, we employed biochemistry, computational dockings, genetic loss of function study, transcriptomics, phosphoproteomics, and pharmacological targeting study has been applied to investigate the fundamental role of the fibrinogens in immune response and neuropathology in COVID-19. And also, we explore the antibody-based approaches to reduce blood clot-related complication in acute and long COVID. We hypothesized that the SARS-CoV-2 directly interact with the fibrinogen, thereby promoting the blood clot formations, altering structure and function of these clots. So our study using solid phase binding assay showed that the fibrin binds to SARS-CoV-2 spike protein. So in this study, to further explore this, we generated custom fibrinogen peptide array. From this work, we identifying the three key binding site within fibrinogen beta and gamma chain. So these sites include in a fibrinogen beta chains, plasmin cleavage sites, and also in the fibrinogen gamma chain, this is the cryptic fibrin binding site for the complementary receptor 3. And these are another area likely formed 3D conformational epitopes in the fibrinogen gamma chain. And the binding mapping study suggesting the multiple point of interaction that could be critical in the context of the clot formation and immune response activation. So using computational docking, we also identified a model indicating close association between fibrinogen gamma chain and the spike ectodomain region here. The further analysis through the alanine scanning mutagenesis, the spike interact specifically with the region in C-terminus of the fibrinogen gamma peptide, which is the critical for binding CD11B-I domain, which play a key role in immune response activations. Our studies also demonstrate that the fibrinogen and SARS-CoV-2 spike protein co-localize in the lung under different experimental conditions. We observed that this co-localization following intravenous co-injection of the spike S1 protein and fibrinogen into the wild-type mice. So using 3D imaging techniques of the solvent-clear organs, including lung in this case, we confirmed that the fibrin and the spike protein, which is the magenta color, interacting within the tissue, suggesting a direct interaction between these molecules in the context of the infection. We next investigate whether spike protein affect polymerization, degradations, and inflammatory property of fibrin. When spike was incubated with a healthy donor plasma, human plasma, in the presence of thrombin, we observed that altered clot structure, increased blood clot turbidity, indicating abnormal fibrin formation. Spike also delayed plasmin-mediated fibrin degradation, suggesting its slow fibrinolysis, contributing to the formation of fibrinolysis-resistant clot seen in COVID-19. Additionally, we found the spike increase fibrin-induced reactive oxygen species production in macrophage. This data highlight that the virus role in amplifying fibrin-induced inflammation. So we examined fibrin-mediated lung and brain pathology after SARS-CoV-2 infection using fibrinogen deficient mice and fibrinogen mutant mice, which expressed mutant fibrinogen that lack CD11b, CD18 binding motif while retaining the normal clotting functions. These mice were infected intranasally with the mouse adaptive SARS-CoV-2 beta variant. We found that in fibrinogen deficients, in fibrinogen mutant mice, there was reductions of the infiltrating macrophage, oxidative stress in the lung, and also suppressed activation of the microglia in the brain. So next, we assess the effect of the fibrinogens on the lung transcriptomes after SARS-CoV-2 infection. So here is the uninfected, and here is the beta strain infected, and left is the wild type, and right side is fibrinogen deficient mice. You can see that the fibrinogen deficiency strongly reduce all the inflammatory pathway, including pro-inflammatory response and interferon signaling pathway, chemokine, ROS productions during SARS-CoV-2 infection. Very interestingly, it also increased genes linked to the NK cell cytotoxicity. So these results suggesting that the fibrin may act as regulator of NK cell during infection. To understand how fibrin suppressed NK cell, we performed the bulk RNA sequencing on the primary mouse NK cell stimulated with the fibrin. Compared with the IL-15, we found that the fibrin significantly downregulated genes involved in NK cell immunity. All the granzyme family and cytokine, chemokines, and reactive oxygen species in the NK cell. Overall, the fibrin strongly suppressed multiple pathways, including NK cell mitochondrial functions, inflammatory response, proliferations, MAC kinase pathway, etc. We also use quantitative phosphoproteomics and kinase activity assay here to study phosphorylation dynamics in human NK cell stimulated with the fibrin or IL-15 as the potent activator of NK cell. Compared with the IL-15, the fibrin suppressed specifically JAK-STAT pathway, multiple the p38 MAP kinase targets, and both of which are key regulator of the NK cell activation. So next, we performed the histological analysis of the NK cell in the lung after SARS-CoV-2 infection. In this study, we used fibrinogen deficient mice and also fibrinogen mutant mice. We observed that increased expression of NK cell marker, NKp46, and granzymes, along with the decreased spike protein expression in fibrinogen deficient and mutant mice. So these data suggest fibrin suppressed NK cell and viral clearance after SARS-CoV-2 infection. So next, we investigated whether harmful effect of the fibrinogen in COVID-19 involved in NK cell suppression. So we infected wild-type mice fibrinogen deficient mice and fibrinogen mutant mice with the SARS-CoV-2 beta variant after depleting NK cell using anti-NK 1.1 antibody. So depletion of the NK cell completely rescue the protective effect observed in fibrinogen deficient mice, and also similarly in mutant mice. With the macrophage infiltration is enhanced after depletion of the NK cell. Also, we found that oxidative stress and viral protein was returning to the wild type levels. So these data suggest that fibrinogen is required for SARS-CoV-2 infection in the lung and pulmonary lesion formation through the inflammatory activation and suppressions of the viral clearance involving NK cell. So the persistent circulating SARS-CoV-2 spike protein has been reported in subset of the long COVID patient. So we examined that the interaction between fibrin and spike could drive thromboinflammation beyond active infection. So using HIV variant pseudotype with the spike protein, we used in vitro and in vivo system with the pseudotype, spike pseudotype variant. In vitro, we found that spike can co-immunoprecipitate with the fibrinogen and also increase fibrin-induced oxidative stress in macrophage. When injected into the mice, spike pseudotype variant caused extensive fibrin deposition and the lung inflammations with increased oxidative stress, which we label with the GP91 FOX, and macrophage activations. Importantly, fibrinogen deficient mice showed a reduced macrophage activations, oxidative stress, these highlighting role of fibrin in mediating this effect. We previously developed the first fibrin targeting immunotherapy. This antibody called 5E8 blocks inflammatory and neurotoxic effect of the fibrin while preserving its normal role in blood clotting. In preclinical model of the multiple sclerosis and Alzheimer's disease, these treatments reduced clinical symptoms and neurodegeneration and harmful inflammation. The humanized antibody version of this anti-fibrin antibody has completed phase 1 safety studies and is currently now tested in the phase 1b Alzheimer's disease and diabetic macular edema. These findings suggest that fibrin targeting therapy could be a promising new approach for neurological disease with vascular damage. We tested the effect of the 5E8 after intranasal infection with the 2 different variants of SARS-CoV-2 model, with and without neuroinvasion. So here we used beta variant, we also used delta variant. And also in published work, we also used a spike pseudotype variant non-infectious model. We performed the histological analysis of the lung and brain after SARS-CoV-2 infection. In the lung, we observed reduced macrophage activations, reduced spike proteins, along with the increase in NK cell marker granzyme expression. In the brain, we found that 5-year treatments decrease in the CD68 positive microglia and loss of the cortical neurons or carbon positive interneuron in the cortex. In addition, transcriptomic analysis of the brain of delta-infected mice show that treatments with the 5-8 significantly reduced expression of the pro-inflammatory cytokine, chemokines, interferon-induced genes, receptor coagulation factors, while the enhancing homeostatic gene expressions. In both the non-neuroinvasive and neuroinvasion model of the COVID-19, in this case, delta infected, we observed that significant fibrin deposition in the brain, consistently associated with the microglia activation in both non-invasive, neuroinvasive models. These findings emphasize that the fibrin play an important role in driving brain inflammation, irrespective of whether the SARS-CoV-2 infection is neuroinvasive or not. We also noticed that after SARS-CoV-2 beta variant infections, upregulation of the disease-associated microglia marker upregulated in the brain, including TREM2, CST7s, compared to uninfected brain. When we use fibrin targeting immunotherapy, prophylactic therapeutic approaches in wild-type mice, our histological data show that 5-8 treatments suppressed microglia activation in the brains. And these findings suggest that the fibrin-targeted immunotherapy effectively suppressed brain inflammation in non-neuroinvasive. And here also found similar effect in neuroinvasive model infected with the Delta variant. And the transcriptomic data analyze 5-8 mediated downregulation of the signaling pathways, also including T cell proliferations, response to signaling activation, chemotaxis, type of interferon productions is also regulated by 5-8 targeting. In summary, the coagulopathy in COVID-19 is not merely a consequence of inflammations, but rather serve as the driver of infections induced thromboinflammation and neuropathology. Also, the fibrin promote neuropathological alteration either indirectly by inducing hypoinflammation through the modulation of NK cell and macrophage in infected lung, or sometimes directly on microglia due to parenchymal deposition in the brain. Our data suggesting that the enigmatic coagulopathy found in COVID-19 is the causal role for fibrinogen in thromboinflammation. And lastly, fibrin targeting immunotherapy may represent approach to selectively suppress COVID-19 pathogenesis in the brain or other organ without adverse effect on normal homeostasis. Lastly, I like to thank my mentor, Karina, and all the members of our labs for their support and helpful discussions. For this study, almost 50 scientists, 5 academic institutes, 2 CRO, and 4 years of collaborations. I want to thank all the collaborators for the valuable contributions and all the funding sources that support this work. Thank you so much for your attention.
Okay, wonderful. Thank you. Sorry, I'm just doing this real quick. Okay, thank you, Dr. Ryu, for presenting for us today. If you have any more questions, just feel free to send them along, and I will get with him and try to get back with you. And then if you could just kind of leave if I don't have your email address, leave it in the chat as well. But if you're signed up with the program, I should have it. So thanks again for joining us. I know it's evening for you. And thanks to everybody else for joining us on today's session. The next session for us is on June 23rd, and the topic that day will be occupational therapy and long COVID, and more specifically, post-exertional malaise, recognizing the crash after activity. Hope to see everybody there, and have a great rest of your day. Thank you
Thank you
again.
so
Thank you,
much.
Dr. Ryu. It was great pleasure meeting with you.
Yeah, pleasure meeting you.
Awesome. Thank you.
Bye.